Hello everyone, my name is Zhiyu Liu. I came from Southeast University. The paper I want to share today is analysis of genome-wide in cell-free DNA methylation, progress, and prospect. I will talk about it from four main aspects: background, method of methylation detection, approaches for cell-free DNA methylation detection, and conclusion. The cell-free DNA in human blood was first reported in 1948, which is made up of short DNA fragments around 160 to 180 bp, corresponding to the unit size of a nucleoson. Recent work has focused on the identification of MOC modifications in cell-free DNA to characterize a variety of potential health conditions. However, the study and application of cfDNA methylation are limited by the fact that the cfDNA in plasma is at very low concentrations and is fragmented. There are many methods for routine DNA methylation detection, and some methods have been improved for the detection of cfDNA methylation. The methods of methylation detection are mainly divided into three kinds: bisulfite conversion, affinity enrichment, and enzyme digestion. Bisulfite conversion is a chemical treatment of DNA that leads to the demyelination of cytosine to uracil. With the high through rate sequences of bisulfite converted DNA, thymidine can be read at unmethylated cytosine positions and cytosine at methylated cytosine positions. Quantification of MOC at individual positions is the rate of methylated cytosine to total cytosine. Bisulfite sequencing has been the gold standard for mapping DNA modifications, including MOC, for decades, and methods based on this principle have been developed. Antibodies may be used to enrich methylated DNA in the protocol. Monoclonal antibodies specific to MOC are used to enrich methylated DNA fragments before sequencing. MDiPSeq can quantify MOC levels at a large fraction of repeats than any other sequencing-based method. The restriction enzyme method is a classical method for the detection of metacytosine. Restriction enzymes are used to cleave single or double stranded DNA on a particular nucleotide sequence. However, the presence of a metal group blocks the cleavage of a particle restriction endomicles. Broadly speaking, two enzymes are used in methylation studies. Methylation-sensitive enzymes recognize specific DNA sequences that are not methylated, while methylation-insensitive enzymes can cleave specific DNA sequences regardless of the methylation status of the recombination sequences. As for the approaches for cell-free DNA methylation detection, they mainly focus on bisulfite-based methods and MIDIP-based methods. Due to the limited amount of available DNA in plasma, conventional methods are poor at detecting cell-free DNA methylation, but some attempts have been made. As the gold standard for methylation detection, methods based on bisulfite conversion have been applied in the detection of cell-free DNA methylation. For example, cancer detection. However, studied bisulfite treatment of cell-free DNA leads to further fragmentation and substantial loss of study materials during desulfonization and purification. In addition, the limiting factor for mutant detection in bisulfite sequence cell-free DNA methylation analysis is the background noise caused by incomplete conversion of bisulfite. Optimizing this step can further reduce background noise. At present, the detection methods require more than 2 milliliters of plasma. Furthermore, the whole gene is amplified with great bias. Thus, efficient protocols for cell-free DNA isolation from plasma and bisulfite treatment are critical factors for the subsequent generation of library of sufficient complementary and successful analysis of sparse amounts of methylate DNA in plasma. Apart from the methods based on biophyte conversion, methods based on affinity enrichment can also be applied for the detection of cell-free DNA methylation for cancer detection and parental diagnosis. However, the limitation of the method in practice is still the amount of DNA in plasma. The usual input DNA for MIDIP-seq is 100 nanograms. However, in Clever combined a liquid biopsy, methylation analysis, and machine learning to develop a sensitive test method based on immunoprecipitation to analyze the material of a limited amount of plasma circulating cell-free DNA, which could detect and classify early-stage cancers. In a word, proof-of-concept studies have shown that cell-free DNA methylation may be a useful biomarker, and there are many prospective studies on the clinical applications of cell-free DNA methylation. However, cfDNA methylation analysis still faces many challenges, the main challenge being that the current method is basically adapted to detect methylation of genomic DNA. More appropriate cell-free DNA methylation assays are needed to increase the sensitivity and specificity of cell-free DNA methylation biomarkers. That's all. Thank you.